So, what kind of blood vessel carries blood away from the heart?
Arteries.
Correct. And what is the big artery that leaves the left ventricle? The biggest one of all. Mirror, mirror on the wall, who's the biggest one of all? Exits.
Aorta.
Yeah, aorta. Okay. So arteries carry blood away from the heart. So as far as blood vessels go, we have different names. So arteries are going to carry blood away from the heart, and veins are going to bring it back. So this is the deal. Basically, we're going to have the aorta, so we're going to systemic circulation, but the same could be true for pulmonary. So we start out with arteries. Arteries are going to get smaller, and we're going to change the name to arterioles. Arterioles, well, they're going to get smaller, and we're going to end up with capillaries, which are the finest of blood vessels. Capillaries are only one red blood cell in diameter. Red blood cells move through capillaries in single file line. So capillaries are microscopic.
And you mentioned those are vessels. Are there any arteries that are capillary size?
These are all blood vessels we're talking about here. Everything on this board. So arteries are larger in diameter and have more layers. Arterioles are thinner. So what we're going here is arterial blood supply. So carrying blood away from the heart. This is the path that blood takes, whether we're talking systemic or pulmonary circulation. We're going to start out with arteries. They're going to start to branch more and more. Arteries will branch and get thinner, and at a certain point, they change their name to arterioles. Finally, arterioles branch out, and we end up with the thinnest of blood vessels, which are called capillaries. Capillaries, so you'll have capillaries in viscera and in all over your body, anywhere you need nutrients, anywhere diffusion is happening. So capillaries, basically all the cells in your body have to be within diffusion distance of blood supply, because you're a big multicellular organism. You're not an amoeba. An amoeba is one cell and it's good to go. It can use diffusion. You can't, you're big. So that's why you have a circulatory system. Blood contains oxygen for your cells to carry out aerobic cellular respiration in mitochondria. Blood also contains glucose. You've heard of the famous blood sugar, and just nutrients for cells. So nutrients are being transported to cells in blood. That is the point of blood. Now your blood itself cannot carry oxygen appreciably. Your blood sucks for carrying oxygen. So your blood has red blood cells in it. They're known as erythrocytes. Erythrocytes are cells full of a protein called hemoglobin. Hemoglobin is the oxygen transport molecule. So that's how oxygen is carried in your blood, is via red blood cells. Your red blood cells don't have a nucleus. They're just basically plasma membranes full of hemoglobin when they're mature. And they have a finite life, and then they're done. Okay, so that's the point. Blood being transported to bring nutrients within diffusion distance of all your cells, as well as oxygen. Oxygen's transported by hemoglobin proteins inside of erythrocytes. Your cells will produce carbon dioxide, and that will get passed back to your blood. Not much carbon dioxide is carried by erythrocytes. Instead, carbon dioxide, a chemical reaction happens in your blood. You'll learn about in physiology. and carbon dioxide is transported a different way, primarily. Okay, so we go arteries to arterioles, to capillaries, to what are called venules. Capillaries are going to converge into venules, and venules are going to converge into veins. So it's kind of like you start out with a big river, and the big river is going to break up into streams, and then the streams are going to break up into creeks, and then the creeks are going to break up into little tiny rivulets of water. Then all the creeks are going to reconverge back into streams, and then the streams join together to form a river again. So that's kind of the way of things. Okay, now let's talk about your systemic circulation, the systemic circuit. Before arteries, there's a major object, a massive artery that has a special name. So what should we start this whole thing out if we're talking about systemic circulation? What's going to precede named arteries, such as carotid arteries and things like that? I'm just rephrasing a simple question that you already answered. Systemic circulation, what's the first big thing that blood's going to travel through out of the left ventricle?
Your aorta.
There it is. So aorta. Okay. So out the aorta, branching into different arteries, branching into arterioles, into capillary beds, into venules. Venules, capillaries will reconverge to venules, converge into veins. So veins are going to pour into two different massive vessels that return blood to your heart. What's the name of those? So veins are gonna, how's blood getting back to your heart? Deoxygenated blood from the systemic circuit enters what? And goes into what chamber of the heart?
The right atrium to the left, to the right ventricle.
Well, we're gonna go to the right atrium. What are the big vessels that return blood to the right atrium? Ignoring the coronary circuit. We're talking just systemic, ignoring the coronary circuit. What are the two big vessels that return deoxygenated blood to the right atrium?
Pulmonary trunk.
Negative. Pulmonary trunk is an artery. It's okay to crack your book open. I'm not going to say anything.
Inferior supravena cava.
Boom, there it is. And I'm going to put the plural because we're going to talk about both of them at the same time. Instead of vena cava or vena cava with an A at the end, I'll slap an E on the end, which is pluralizes it. Maybe it's vena cava, I don't know. I'll just leave it like that. Okay. So that's it. That's systemic circuit. So let's talk about the pulmonary circuit. Pulmonary circuit. All right. So what's the first thing for the pulmonary circuit?
Pulmonary trunk.
Boom. Okay. Pulmonary trunk. Pulmonary trunk. Okay, we're coming on back now. Veins. Okay, well, right and left, superior, inferior pulmonary veins. That's it. Bam. And then we're going to go to the left atrium. Okay, so on the pulmonary circuit, is the blood heading out the pulmonary trunk to the pulmonary arteries? Is that blood oxygenated or deoxygenated?
Deoxygenated.
Correct. The blood returning via the right and left superior and inferior pulmonary veins, is that oxygenated or deoxygenated?
Oxygenated.
For sure. Beautiful. Okay. So let's talk about the structure of arteries and veins. So we're going to have three tunics, cloaks in general, but things are going to change. We're talking about capillaries, because capillaries are just like epithelial cells, and that's it. Okay, so we have for blood vessels, we have the tunica. Let me switch this pen, it's dying. Okay. So the three tunics. Ooh, that's a nice one. We have the tunica intima, is the innermost. Then we have the middle layer, is called the tunica media. And then we have the external layer, or the superficial layer, where you're going from out to end, the tunica externa. So this is the structure of blood vessels. There's going to be some differences in thickness of the layers and a couple extra features between veins and arteries. As we go smaller and smaller and move out into those little creeks, capillary beds, they're going to lose some of the tunics. Okay, so the tunica intima is the innermost. It's called endothelial, which is just inside epithelial. So there's the endothelial layer, simple squamous. So the tunica intima has an endothelium. That's the innermost layer. So the blood's moving across simple squamous epithelium, which is the best kind of cell or tissue for diffusion. It is the best. Flat and perfect for diffusion. Now, for vessels that are a little larger than a certain diameter, I forget the exact number, but we'll see on the slideshow, there can be a subendothelial layer. It's just a little bit of connective tissue. We can also have a subendothelial layer, which is just CT. Okay, the tunica media is smooth muscle. So the middle layer, the tunica media, is smooth muscle. The tunica externa is CT. connective tissue, the outermost layer. So these are the tunics. Now, if we're looking at arteries, arteries have a thicker tunica media than veins. Arteries have more muscle because arteries, and another thing the arteries have is more, they have elastin, they have elastic. Arteries are elastic because they need to deal with high pressure blood, and arteries have like pulsatile flow. So arteries need to be more elastic because they get stretched from high pressure blood and then they snap back. So in the tunica media, the smooth muscle of arteries, there can be a considerable amount of elastic fibers. Additionally, they might have these couple extra layers, like an internal and external elastic sheet. So there can be some differences between different types of arteries, and definitely differences between veins. In general, arteries are thicker walled than veins. So for the tunica media, arteries can have two sheets of elastic membrane. Let's just say two sheets of elastic for now, not called a membrane. So some of the arteries, they'll have a layer of elastic, I'll say membrane, but membrane is a poor choice of word. Basically, the tunica media, the smooth muscle, can be sandwiched. So you have a big, thick slice of meat and two pieces of bread on either side. So that can happen for arteries. So it'll be the tunica intima, the endothelium and subendothelial layer for thicker arteries. Then there'll be an elastic sheet, smooth muscle, another elastic sheet, and then we go to the tunica externa. Okay, let's see if this gosh darn slideshow is working now, so we can see some pictures of this stuff. Ooh, it opened just like that. Boom. Right. Okay, types of blood vessels. Three major types: arteries, capillaries, veins, but I give you more. Okay. Arteries carry blood away from the heart. Arterioles, smaller arteries. Capillaries, the smallest blood vessels of all, converge into venules, which converge into veins. Okay, three layers. Tunica intima, the innermost tunic, contains the endothelium, simple squamous epithelium. Vessels greater than 1 millimeter also have a subendothelial layer. You'd think I would remember that, 1 millimeter, it's an easy number, but I don't. Okay, the tunica media, sheets of smooth muscle. Okay, tunica media is the middle layer. Tunica externa, connective tissue. Now there's a couple terms we'll come back to. Vasoconstriction, vasodilation. Basically, vasoconstriction is a narrowing of a blood vessel. Vasodilation is a relaxation of the blood vessel. So vasoconstriction, they get narrower, vasodilation, they get wider. That's going to be for arterioles where this happens. That can alter your blood pressure and also alter blood flow to an area. Okay. Oh yeah, the lumen. That's just the central space of the blood vessel. What do you think's moving through the lumen? Oh, it says right there it's blood. Okay. Now, here's something weird. The largest blood vessels, like big ones, they can have something called vasa vasorum. What these are is vessels of the vessels. The blood vessel is so big that in the tunica externa, there's actually little tiny fine blood vessels to bring nutrients to the blood vessel itself. The inner region, there's blood moving through it, it's going to get its nutrition there. It's not like the heart. The heart, blood moves through the heart. The heart isn't using that. The heart has to have its own blood supply separate. For blood vessels, they are going to pick up on nutrients. They're going to nourish themselves with the blood moving through them. But when they're really big, they need an outer blood supply because it's too far for nutrients to get to the outer parts of the blood vessel. So that's vasa vasorum, vessels of the vessels. So here we go, the generalized structure, and this is what I was wanting. The tunica intima. Okay. The endothelium, simple squamous epithelium. Whether we're talking veins or arteries, same thing. Now, for vessels greater than 1 millimeter in diameter, we're going to have a subendothelial layer of connective tissue. Or I guess a basal lamina, something like that. Okay. Now, the tunica media is going to be smooth muscle, and there can be elastic fibers. Now, what's being shown here in this picture, the artery, is a special kind of artery. A distributing artery, I believe. So having this internal elastic membrane and external elastic membrane, only one type of arteries have this structure. So membrane is a good word. Internal and external elastic membranes. Sandwiching the tunica media. Looks like Swiss cheese here in the diagram. I said it was bread, but it looks like Swiss cheese. So you went and got a can of Spam, sliced off a big piece, and just slap Swiss cheese on top and bottom. Yum. My favorite, bitter Swiss cheese and Spam. Might as well just up it a level and make a lasagna with some 9 lives straight out of a can. Okay. Tunica externa, connective tissue, and here you see those vasa vasorum. This little piece of poo-poo, when I restarted it to make the slideshow work, it decided it doesn't like the Apple Pencil. Which means I have to restart again. $1,000, brand new iPad and Apple Pencil. You think it would work, right? Should I call and complain? Well, we'll keep on looking at this. Okay, so here you see vasa vasorum. Now, can everyone remember vasa vasorum? Come on, Alexis, you can remember it. Vasa vasorum, vasa vasorum, vessels of the vessels. That's it. That's all there is here. Now look at the vein on the right. Right off the bat, you notice the tunica media is thinner. Also, you'll notice there is no internal or external elastic membrane. And veins have far less elastic fibers in their tunica media, period. Blood moving through veins is low pressure. also notice in the vein that the vein has a much thicker tunica externa. That's not immediately apparent from this diagram, but veins have a larger connective tissue covering and a thinner smooth muscle layer. So thicker tunica externa in veins, thinner tunica media. I see one other structure on the vein that the artery lacks. What is it? Do you notice anything in the vein that the artery does not have? Or does the vein have, or the artery lacks that the vein has? What is it?
The valve.
Correct. That's it. So veins have valves, and we'll talk more about those later. Now look at the capillary. Do you see all these tunics for the capillary? At the bottom, what do you see for the capillary?
Cells.
Huh?
Endothelial cells.
That's it. So all it has is a tunica intima and then a basement membrane, underlying membrane around the simple squamous epithelial cells. That is a capillary. Capillaries are built for diffusion. Now we look at the left, the artery. Okay, so we have the artery. Then move out to the right, it's red and smaller than the artery. Moving into the, it's labeled capillary network. That's also called a capillary bed. Capillary bed. Okay, so the little small red colored object moving into the capillary network here in the picture, but it's a capillary bed. What is the name of that thing? The little small red vessel coming out of the artery. What would you call that?
Arterioles.
Yeah, arteriole. Okay, so we go from the artery, the arteriole, into a capillary bed. And then the capillary network in the capillary bed is going to reconverge into what structure? What's the name of it?
Venules.
Venule, and back to the vein. Cool, so it's got it all right here. Okay. All right, well, let's talk about arteries first. We have different ones. The biggest ones are called conducting arteries, also known as elastic arteries. Okay, before I do that, what are the differences between this artery and vein?
Arteries thicker.
Thicker wall, yeah? What is thicker most of all in the artery?
The vasa vasorum.
No?
The tunica media.
Tunica media. What about the lumens of the two? Which one has a larger lumen?
The vein.
Yeah. So veins have larger lumens, thinner tunica media, less elastin, less elastic fibers than arteries. Okay. Oh, and as we go throughout your body, veins usually travel right alongside arteries. So blood's going to go to a place with arteries, and blood's going to come back from veins. But they're going to be side by side. Veins can pick up on some of the pulsatile flow of the artery. The pumping of the artery actually helps move blood through the veins. And there's some other ways blood gets moved through the veins also. Valves, for one, help prevent backflow in veins. Okay, describe the three tunics, define vasa vasorum, compare and contrast arteries, capillaries, and veins. Okay, types of arteries. Number one, elastic arteries, the largest. Diameters, 1 centimeter to 2.5 centimeters. The aorta and its major branches is an elastic artery. They're also called conducting arteries. They're there for high pressure blood to just move through them. So they have a lot of elastin to deal with this high surge of blood pressure. So this is elastic arteries. Conducting arteries, same thing. Aorta and a couple others. Next, we're gonna have, okay, well, let's look at a picture. An elastic artery, okay. We see the tunica externa with vasa vasorum. We see a lot of elastin in the tunica media, and then we see the tunica intima. Ironically, if we wanted to see elastic CT, we would look at a cross-section of the aorta. Since we didn't have that at Fallbrook, we never got to see elastic connective tissue. But we would have had to slice through the aorta to take a look. Okay, so that's this, thick tunica media with tons of elastin. Next up, distributing arteries, also known as muscular arteries. Most named arteries are distributing arteries, muscular arteries. Where are they located? Distal to elastic arteries. Okay, thinner in diameter. Tunica media is thick. The unique features, internal and external elastic laminae. Okay, that's the word I like, not membrane. Lamina, that's the word I was looking for. Whatever. Sheets, elastic sheets. Spam sandwich. So that's the unique feature. So now we know, looking here, that we are being shown a distributing artery. That's a muscular artery, AKA distributing artery. Because we see the internal and external elastic laminae. Right. Here's a picture. Muscular artery, tunica externa, tunica media with an external elastic, gosh darn membrane again, external elastic lamina and internal elastic lamina. Lumen. Arterioles, we're going to start to drop off some tunics. Smallest arteries, 0.3 millimeters to 10 micrometers. Larger arterioles possess all three tunics. Smaller are pretty much just a single layer of smooth muscle with underlying endothelium. When we move to capillaries, they're going to ditch the smooth muscle. Capillaries are designed to be leaky. They leak because nutrients need to get out to your cells where capillary beds are located. So arterioles, smallest arteries. Larger ones, all three tunics. When we move down to narrower, smaller arterioles, we're going to drop a tunic. Which tunic's being lost? What two tunics are going to be present in the smallest arterioles, or which tunic is lost?
The intima?
Negative. Hey, you're trying.
Externa?
Yes, tunica externa is gone. We still have the tunica media, just a single layer, and we have the tunica intima. Okay, when we get to capillaries, okay, oh gosh, it's just going on and on. Capillaries, all we have is the tunica intima and a basement membrane, that's it. Smallest blood vessels, 8 to 10 micrometers. Microscopic. Red blood cells, erythrocytes, move through in a single file line. What are capillaries good for? Diffusion. They're king. Simple squamous is the king of diffusion. Out of all the epithelial cells, that is the one. Simple squamous is the best for diffusion. Okay, so capillaries. You have site-specific functions of capillary beds. In general, capillary beds are leaky. You lose blood plasma out of capillary beds, not formed elements, not cells. Blood cells are not going to move out, but a lot of fluid gets leaked out. Nutrients readily leak out. We have different types of capillaries too, 3 different kinds. We'll see those shortly. But you have site-specific functions. In your lungs, in the pulmonary circuit, you have pulmonary capillary beds are all around your lungs. Why? Because oxygen is going to diffuse in to your blood. CO2 is going to exit in your lungs. Your small intestines, nutrient absorption. Your small intestines those are the sites for absorption of most of your nutrients. Your large intestine only reclaims water, that's it. And a few vitamins that bacteria make there. So small intestines, capillary beds pick up all your nutrients. In your endocrine glands, capillaries pick up hormones to distribute throughout your body. In your kidneys, capillaries in your kidneys, that deals with filtering your blood, not for pathogens, but for waste. Nitrogenous waste. Anyone ever heard of urea before?
Yeah.
Yeah, so urea, basically, when you break down amino acids, protein, the nitrogen that's in an amino acid, you don't need it. So it's going to be nitrogenous waste. It needs to get out of your body. So it'll leave as urea. If you were a fish, you would just excrete ammonia. But you're not, you're a mammal, so you excrete primarily urea and a little bit of a couple other things. Okay, so capillaries, they're in your kidneys because you're going to filter your blood for waste. Your blood gets adjusted. The composition of your blood is adjusted in your kidneys. Your endocrine glands, hormones will be put in your blood for target cells, chemical message. Small intestines, you need nutrients to go in your blood supply. Lungs, you need oxygen, you need to ditch carbon dioxide. There you go. Red blood cells, erythrocytes, move in single file through a capillary. Cool. Okay, let's go back. Okay, so we just took it down from conducting or elastic arteries like the aorta and its major branches. We took it on down to muscular arteries, also called distributing arteries, most named arteries. And then we went into arterioles. The smallest ones are just a single layer of smooth muscle in the tunica media, plus the tunica intima. And then finally we went to capillaries. Okay, so more on arterioles. Arterioles, that's where your blood pressure is regulated. Okay, so we have vasoconstriction and vasodilation happens. So arterioles, they can constrict and get smaller in diameter or relax and get larger in diameter. The word is vasoconstriction. Vasodilation, they widen. What part of the nervous system, if any, is responsible for vasoconstriction?
Sympathetic.
Sympathetic. What about the parasympathetic? Does it do anything?
Dilation.
Doesn't do a thing. Your arterioles are controlled 100% by the sympathetic. Nobody else. That's it. So when the sympathetic signal stops, presumably they relax. However, local tissues where blood's going to, they can release chemical messengers. Local control tissues can release chemicals that stimulate arterioles to constrict or dilate. So the sympathetic controls it, and tissues might control what blood's coming to them. If they don't need anything, maybe chemicals there cause vasoconstriction. They need more blood. Some chemicals from those tissues will be released to induce vasodilation. What neurotransmitter would the sympathetic use from the synaptic terminals and the postganglionic neurons to stimulate vasoconstriction? Remember, there's a 2-neuron chain for the sympathetic: preganglionic, postganglionic. The ganglia is outside the CNS. It's a collection of neuronal cell bodies. We always have a 2-motor neuron chain for the autonomic nervous system. So we're talking the postganglionic neuron, the second one in the chain. For the sympathetic, what neurotransmitter will it use to initiate vasoconstriction?
Norepinephrine.
Gotcha. Nope, acetylcholine. Gosh darn it, there goes all my test questions. I'm losing test questions by the minute. The sympathetic alone innervates what? Most things, like your heart, has sympathetic and parasympathetic control, right? The heart has its own pacemaker with the sinoatrial node. However, the vagus is the parasympathetic, can slow your heart rate down, beat, under direction of the medulla oblongata. The sympathetic division will speed your heart up. So both work on the heart. Same was true of the lungs, things like that. Both work on the iris in your eye. However, the sympathetic has sole jurisdiction over 2 things, name them. We just talked about one thing they control. Are you looking at it on the screen? Arterioles, right?
Yeah.
Does the parasympathetic play with it? No. What's one other structure the sympathetic alone controls? I'm not going to tell you. Just think about that. Okay. Sympathetic doesn't need to play with norepinephrine when it has sole jurisdiction. Okay. Arterioles. They regulate your blood pressure by changing their vessel diameter. Diameter of arterioles controlled by local factors in the tissues, so releasing molecules, saying, hey, we need more blood, or hey, quit feeding us, we're full. Or the sympathetic. Vasoconstriction, vessel narrows, blood pressure goes up. Vasodilation, vessel widens, blood pressure goes down. Okay, let's look at arterial wall structure. Here's a small arteriole. You see it possesses a single layer of smooth muscle, so tunica media, then it has an endothelium and lumen, and that's it. The smallest of arterioles. There you go. Compare and contrast this, this, and this, and this. Capillaries, we started talking about them. Red blood cells move through single file. There they are. Aren't you the slightest bit curious about blood in general? The different cells in your blood? Are you curious or you're not curious? I would be. Blood's one of the coolest things to talk about out of any of this, but we'll see. Okay. Where'd everybody go? The screens are all dark. I see Ashley, Alexis, Casey, everybody else disappeared.
Is it the skin?
Huh?
The sympathetic?
Kinda.
Okay.
What in the skin? Okay.
Where?
All right. You got to be more specific. Okay. What exactly in the skin? All right. I'm not saying. Okay. So we have different types of capillaries. Let us take a look at something else real quick. And let me restart this so that Apple Pencil works. We're going to look at something else and we're going to take a quick break and have a word from our sponsor. Oh, there's Megan. She's back. I don't know. I thought Megan left for a bit and put a stuffed animal in front of the camera. She's like, here, talk to my kids' stuffed animals for the next hour. Okay, let's have a look at this. What is it? Not ready yet. Okay, let me see. Are you ready for this action?
Yes.
Okay, boom. All right. Bam. Ooh, look at that. Beautiful. Okay. What's that called? Tunica externa. All right, it's labeled. Gosh darn piece of junk. What are all these little red and blue things sticking out here that are located in the tunica externa? You see them right here too. See them in the tunica externa of the vein?
The vasa vasorum?
There it is. Boom. Okay. I see 2 sheets of Swiss cheese right here. They're sandwiching what in the artery? What's that big piece of Spam in between the 2 pieces of Swiss cheese? Okay, what's the outer one? The outer Swiss cheese, the more superficial, directly deep to the tunica externa.
Tunica media?
Okay, so the Spam is the tunica media. All right. So what's this outermost Swiss cheese called?
External elastic membrane.
Sure, or lamina. Okay. And the other one's the internal elastic lamina. Cool. We have it all. Vasa vasorum, all of it. Okay. We look at the vein, we even see a valve right there, venous valve. I'm wondering what this is. So this is the endothelial layer right there, vascular endothelium. Okay. So that's the tunica intima. This must be the subendothelial layer. Now it's the basement membrane. So of course the endothelial layer has to have a basement membrane because all epithelial tissue does. But what's this? Subendothelial layer. All right. So, bam. Squamous epithelial tissue, basement membrane, subendothelial layer, which means that this vein is greater than 1 millimeter in thickness. So it's going to be the same thing over here. Okay, the endothelial layer, the basement membrane, and the subendothelial layer. Okay, what's this structure called? The first one that's leaving the artery. Okay. Arteriole. What do we have here? The capillary network, capillary bed. What's there? Venule. This is a badass model, isn't it? I think it's pretty dope. It's awesome.